Hello, everybody. Welcome to today's Revive webinar by the Global Antibiotic Research and Development Partnership, GARDP. My name is Astrid Pence-Moore, and I'm hosting this webinar on intracellular models for antimicrobial R&D. For those of you who don't know Revive yet, Revive is GARDP's education and outreach program, and it aims to connect and support the antimicrobial R&D community by facilitating learning, sharing knowledge, and connecting people. The Revive webinars are part of our educational activities. All webinars are recorded in full and can be viewed after the live broadcast on our website, revive.gardp.org/webinars. And I would like to encourage you all to visit the Revive website to stay up to date about future webinars, watch recordings of previous webinars, and also find other information. As usual, today's presentation will be followed by a Q&A session. You can submit your questions at any time during the webinar via the questions window in your webinar control panel as shown on this slide. We will address the questions after the presentation, and we will do our best to respond to as many questions as possible. Today's speaker is Françoise van Bambeke, and my colleague Laura Piddock, Director of Scientific Affairs at GARDP, will moderate this session. Welcome, Françoise. Laura, I'm handing over to you.
Thank you, Astrid. I am very happy to welcome our speaker, Françoise van Bambeke. Françoise is a research director from the Belgian Fonds de la Recherche Scientifique. She is a professor of pharmacology and pharmacotherapy and carries out research in cellular and molecular pharmacology at the Louvain Drug Research Institute at the Université catholique de Louvain in Belgium. Françoise received her PharmD and her doctorate in pharmaceutical sciences from the Université in 1991 and 1995 respectively. In 1997, Françoise carried out postdoctoral research in molecular biology of drug resistance under the supervision of Patrice Corvalin at the Institut Pasteur in Paris, France. She became a professor in 2008 and received a full professorship at the Université catholique de Louvain in 2013. She has several research interests, including cellular pharmacokinetics, pharmacodynamics of persistent infections, cellular toxicodynamics of antibiotics, and the search and pharmacological evaluation of new antibiotics directed against new targets. Françoise is a member of numerous national and international scientific societies, and she is on the editorial boards of several scientific journals. These include Chemotherapy, Journal of Antibiotics, and Antimicrobial Agents and Chemotherapy. Welcome, Françoise. Please start your presentation.
Thank you, Laura, and thank you, Astrid, for the kind introduction. So I'm very happy to be here today, and I hope you will enjoy this presentation about intracellular model for antimicrobial research and development. So these are my disclosure for this presentation. So as you can see, I received research grants from many companies over the last years, mainly for the study of antibiotic pharmacokinetics or pharmacodynamics against intracellular infections. So this is our agenda for today. So I divided my talk in 4 parts. So first of all, we will review briefly the role of intracellular survival in chronic infection and its contribution to poor response to antibiotics. Second, we will look a little bit how to develop in vitro models to study intracellular activity of antibiotics. And then we will review the cellular pharmacokinetics and pharmacodynamics parameters that can be considered as predictive of intracellular activity for antibiotics. So let's start by briefly reviewing how bacteria could be normally killed by phagocytic cells in our organism. So when a bacterium comes in the contact of a phagocyte, normally it will be phagocytosed and driven in a specific vacuole called a phagosome. This phagosome will then fuse with lysosomes to form phagolysosomes. And in these vesicles, the bacteria will be exposed to several host defense mechanisms that will be able to kill the bacteria. However, several bacteria were able to develop mechanisms to avoid this destruction and therefore survive inside eukaryotic cells. And you can see here a few examples of these bacteria surviving within the cells. So there are well-known examples like Mycobacteria, which are found in vesicles closely related to endosomes, Listeria monocytogenes or Shigella flexneri, which will thrive in the cytosol, Legionella pneumophila, which is able to survive in two types of compartments, either the endoplasmic reticulum but also in the phagolysosome, Staphylococcus aureus, mainly found in the phagolysosome, Salmonella and Brucella in phagosome, and finally Chlamydia in inclusion. So you can see that virtually we can find bacteria in all subcellular compartments. So the question we may ask now is, what are the benefits for a bacterium to survive within an eukaryotic cells? The image I would like to use here is the one of the Trojan horse. And this image illustrates for me three main advantages for the bacterium to survive within an eukaryotic cell. The first one is that it will help the bacteria to invade the host. The best example for this is probably the invasion of the central nervous system by Listeria monocytogenes. So several models have been proposed to explain how the Listeria is able to travel from the gut to the central nervous system, and you have here a summary of the main route it can follow. So it has been proposed, for example, that Listeria can travel using the bone marrow monocytes. Other people describe also that it is able to adhere and then to be transferred from the monocyte to the endothelial cells. And finally, it has also been described that the Listeria can be directly found inside the axon of the neurons. A second advantage of the intracellular life is that it can help the bacteria to persist within their host. Here is an example of this situation, which shows in electron microscopy the presence of Staphylococcus aureus in osteocytes, as you can see here, but also in osteoblasts, like here, and finally also in the bone matrix of bone from a patient suffering from a recurrent osteomyelitis. And finally, the third advantage of intracellular life is that it will protect the bacterium from the activity of antibiotics and also of host defenses. And of course, this is the main part of the topic of today. So this slide illustrates by a few examples that we are facing therapeutic failure when we wish to eradicate intracellular bacteria with antibiotic. And during this presentation, we will try to review the parameters that can explain the loss of activity of antibiotics against this very specific form of infections. Let's now move to the second part of the presentation, which will describe in vitro models to study intracellular activity of antibiotics. So a series of models have been described in the literature, but today I decided to present the one we developed in my laboratory, which is in fact a quite classical model. So how should you do if you wish to develop an intracellular model of infection? So the first step consists in opsonizing the bacteria in the presence of human serum. So this step will allow to cover the bacterium with specific substances that will help the bacteria to adhere to the cell surface. Then you put these opsonized bacteria in the presence of your cell of interest, like for example here an adhering macrophage. And an important step here when you develop the model is to select an appropriate multiplicity of infection, meaning an appropriate number of bacteria per eukaryotic cells. So this is quite critical because if your inoculum is too low, you will never be able to infect the phagocytes, and if your inoculum is too high, you will kill the cells because, of course, most of these bacteria can exert a certain toxicity on the cells. Then you let these bacteria in contact with the cells for a given period of time to allow their phagocytosis. And then, of course, you need to eliminate the bacteria that have not been taken up by the cells or that are still adhering at the cell surface. And to this effect, the best strategy is to use the antibiotic gentamicin at a very high concentration. So why gentamicin? So gentamicin is an aminoglycoside antibiotic, and this antibiotic is in fact characterized by a very rapid bactericidal activity, so meaning that it will very rapidly kill the bacteria in the culture medium, and at the same time, this antibiotic will also enter the eukaryotic cells only very slowly. So this means that if you do a short incubation with a high concentration of gentamicin, you will not allow the antibiotic to enter inside the eukaryotic cells, and therefore you will not have any influence on your experiment. Then the next step will be, of course, to eliminate gentamicin by an appropriate washing, and then to incubate the infected cells with the antibiotic of interest for your study. After a given incubation time, you will then lyse the cells in order to recuperate your bacteria, and you will plate, of course, an inoculum on an agar plate in order to be able to measure the CFU on the next day. In parallel, you also need to normalize your experiment, so meaning to try to consider a similar number of infected cells, and for that you can, for example, measure the protein content of your sample, which is an indirect measure of the amount of cells you have in your sample. So using this technique, this is typically the type of results you can obtain. So in this graph, you can see here the extracellular growth of Staphylococcus aureus, so the growth in a broth, for example. And in red, you can see the intracellular growth in G774 mouse macrophages. 0 represents the initial inoculum, so meaning the inoculum at the end of the phagocytosis period. So as you can see here, extracellularly, the bacteria will grow all over the time, but intracellularly, you have first a sort of latency period during which the apparent multiplication rate of the bacteria is close to 0. After this period, the bacteria will start also to multiply. If you look to this cell in the electron microscope, you can see here that just after phagocytosis, the bacteria are well visible, isolated in a vacuole. So you may see here the membrane of this vacuole. And if you look at the end of the experiment, you can see that the bacteria indeed multiplied inside your cells. And if you look carefully, you can see here, for example, that they are still located in vacuole, which are in fact phagolysosomes. So once you have developed this type of model, as I said, you can expose them to antibiotic. This is an example of macrophages infected by Staphylococcus aureus and exposed to the fluoroquinolone moxifloxacin. So in this experiment, you can see on the top the activity against extracellular bacteria, so meaning bacteria growing in broth. And on the bottom, you can see the activity of moxifloxacin against intracellular bacteria. So if you look in the literature, when people do this type of experiment, they usually select a few number of concentration. So for example here, a concentration equivalent to the MIC, 10 times the MIC, or if you wish to have a more clinical relationship, you can also use a concentration mimicking the human Cmax, so the maximal concentration you can find in human serum. And then you follow the activity over time. And what you can see here very easily is that for the 3 concentration investigated here, the activity is much lower against intracellular bacteria than against extracellular bacteria. So for example, if we take the human Cmax, you can see here that after 24 hours, we reach almost the limit of detection, -5 log, while against intracellular bacteria, we only reach a reduction of 2 logarithms. So this is already interesting, but in fact, the type of information you can extract from this experiment is limited. If you really wish to explore in detail the pharmacological profile of the antibiotic, you need to go one step further and to do another type of experiment. And this is what is illustrated here. So in this experiment, we did in fact the same type of thing, except that instead of selecting a very few number of concentration, we exposed the bacteria to a very wide range of antibiotic concentration, including concentration that are well below the MIC, so supposed to be inactive, and concentration which are markedly more elevated than the MIC. The idea when doing this type of experiment is to be able to draw what we call a full concentration response curve, as you can see here. And with this type of response curve, we will be able to extract some useful pharmacodynamic parameters that will allow us, for example, to compare different drugs. So if we look globally, you can see here the curve for the extracellular bacteria and the curve for the intracellular bacteria. And again, it is very clear that your activity is much lower against intracellular bacteria. So as I said, using this curve, you can establish different pharmacodynamic parameters. And for that, you will use the equation of this relationship, which is in fact a sigmoidal dose response, or also called the Hill equation. And so using this equation, you can calculate a first useful parameter, which is the static concentration, abbreviated here as C-stat. So what does that mean? So the static concentration is the concentration of antibiotic that you need to obtain a bacteriostatic effect. So remember that this 0 value here is the initial inoculum. And you can calculate this value, for example, here for the extracellular activity and for the intracellular activity. And on a pharmacodynamic point of view, the static concentration of a drug will tell you about the relative potency of the drug, meaning it corresponds, in fact, to the MIC of the drug in a specific environment, either extracellularly or intracellularly. And indeed, you can see that these values are very close to the MIC of the antibiotic. The second parameter, which is of high interest, is what we'll call Emax, or the maximal efficacy. So how do you calculate this parameter? In fact, the maximal efficacy corresponds to the maximal reduction in inoculum you can get for an infinitively high concentration of antibiotic. It corresponds, in fact, to the plateau that you can obtain here at the end of your dose response. And again, you can calculate this value for your intracellular antibiotic or for your extracellular antibiotic. And as you can see here, the value is much more negative against extracellular bacteria than against intracellular bacteria, meaning that the antibiotic is much more effective against the extracellular bacteria. So to sum up about these parameters, so what do they tell to you? So regarding the relative potency, it can be considered as an estimation of the concentration you need to reach a specified effect. In fact, it can be considered as a measure of the intracellular MIC, and therefore this parameter is mainly related to pharmacokinetics because it will depend on the accumulation of the antibiotic in the infected compartment, but also it will also depend on the intracellular bioavailability of your drug. This parameter will also be influenced by the local environment inside the cells because this environment can influence the intrinsic activity of the drug. For example, the pH can be different from one compartment to another one. So for example, in the phagolysosome, the pH is acidic, and we may also have an interference with the oxidant species present in phagocytic cells. So in most of the cases, what we will observe is that this parameter cystatic is higher intracellularly than extracellularly, higher or equal. Regarding now the maximal efficacy, so this parameter is rather an estimation of the maximal reduction in inoculum for an infinitively large concentration. So this parameter will in fact help you to evaluate the killing capacity of your antibiotic. So this is a pharmacodynamic related parameter. It will depend on the mode of action of your drug, of the bacterial responsiveness, and also of the capacity of the antibiotic to cooperate with host defenses. And here, in most of the cases, the maximal efficacy is much lower intracellularly as compared to what you can measure extracellularly. So this slide summarizes the different pharmacokinetic and pharmacodynamic parameters that we need to take into account to evaluate the intracellular activity of an antibiotic. So let's first focus on pharmacokinetics, and these parameters are in green on this slide. So first of all, you need, of course, to have an antibiotic which is capable to enter inside the eukaryotic cells and for which also efflux is minimal. Then you need also to take into account a possible metabolism or degradation of your antibiotic inside the cells, as well as a possible binding to some specific cellular constituent. And then finally, the important parameter for related to activity will be the accumulation of the antibiotic inside the cells and its bioavailability, so meaning its availability to exert its activity. Focusing now on pharmacodynamics, so these parameters are presented in blue, we have to take into account three main parameters. So the first one is the bacterial responsiveness. So what does that mean? It simply means that a bacterium, when it is located inside a cell, it will not respond to your antibiotic as in a bacterial broth. And this is very logical because a bacterial broth is a medium that has been optimized to favor bacterial growth. And of course, this is not the case of the intracellular medium. A second parameter is the physicochemical condition prevailing in the infected compartment. So as I said before, the best example is the one of phagolysosomes. So in this organelle, the pH is acidic, around 5.5. And of course, this can influence the activity of the antibiotic, but also possibly the bacterial response. And finally, we have also to take into account the cooperation with the host defenses, which can be very active, for example, in professional phagocytic cells. Let's now move to the third part, where we will examine the cellular pharmacokinetic parameters that can predict the intracellular potency for your antibiotic. So before looking to these parameters, let's first examine how the antibiotic can enter inside eukaryotic cells. The first mechanism is by simple diffusion. And this is illustrated here. So you have your drug in the extracellular medium, and it can simply diffuse through the cellular membrane, which means not only against the pericellular membrane, but also through the membrane limiting the different organelles inside the cells. So in general, when the antibiotic enters the cell by simple diffusion, its concentration inside the cells should be equivalent to the extracellular concentration, except if there is some binding site inside the cells which can retain the antibiotic. Second, when the antibiotic enters by simple diffusion, its cellular concentration is in principle linearly related to the extracellular concentration. So this is represented here. So if you expose the cells to different extracellular concentration, the ratio between the concentration inside and outside, which is the accumulation, will remain constant. Diffusion is in principle a very rapid process. And this is illustrated here, so you can see that it can be very rapid till reaching a specific equilibrium. And second, the rate of efflux should be equivalent to the rate of efflux because the diffusion can operate in both directions depending on the gradient of concentration. In principle also, as I said before, an antibiotic entering the cell by diffusion has probably access to the different compartments because it can also diffuse through the intracellular membrane. So one example of antibiotic following this mechanism for entry inside eukaryotic cells are beta-lactams. So in the literature, the people will often consider that beta-lactams do not accumulate inside eukaryotic cells, and therefore in many textbooks you will find that they will not be active against intracellular bacteria. So this is not totally true because beta-lactams are able to diffuse through the membrane of eukaryotic cells, so they will enter inside the cells, but they will not accumulate. So it means that their cellular concentration is equivalent to the extracellular concentration, but as you know, the circulating concentration of beta-lactams are usually very high, and so it means that intracellularly, you can also reach concentration large enough to act upon intracellular bacteria. A second mechanism that we can observe is the presence of transporters. And you can have, again, transporter in both directions. So either you can have an influx transporter, and in this case, it will favor the entry of the antibiotic, but you can also have efflux transporter, and in this case, it will increase the efflux of the antibiotic. So what can we comment about these transporters? So if you have an influx transporter, your accumulation will be usually more rapid than by simple diffusion. And second, your accumulation will usually be lower for higher concentration because you can have a sort of saturation. So this is represented here. So for low concentration, your transporter will be helpful to help the drug to enter the cells, but at high concentration, you will saturate the transporter, and therefore it will not be able to allow the drug to enter in the cells. If you have now an efflux transporter, you will have, of course, the opposite situation. So the efflux will be more rapid than by simple diffusion. So this is illustrated here. So the efflux is more rapid than by simple diffusion. And second, the accumulation in this case will be higher for higher extracellular concentration. Why? Simply because in this case, you will saturate also the efflux transporter, but if you saturate the efflux transporter, you will keep more drug inside the cells, and therefore you will increase your accumulation. So typical efflux transporters that can play a role for antibiotics are, for example, the P-glycoprotein that you may know, and also other types of multi-drug related resistance proteins. One example of drug which is substrate for an efflux transporter are fluoroquinolones. Let's move to the third mechanism, which is endocytosis. So this mechanism will be used by antibiotics which are unable to cross the eukaryotic cell membrane. And this will be the case for drugs that are very polar, like aminoglycoside, for example, but also for drugs which have a very high molecular weight, like glycopeptides, for example. So these drugs, therefore, are unable to cross the membrane. And so for them, the only way to enter an eukaryotic cell is endocytosis. So they will be taken up like a bacteria, as we saw before, in a vesicle. And within this vesicle, they will be driven in the phagolysosome, which is an acidic compartment. So in contrast to diffusion, this process will usually lead to a higher accumulation, especially if it is mediated by a binding at the cell surface, because this could correspond to a low affinity transporter. It could also, therefore, be saturable for high concentration. Second point, this process is usually slower than diffusion. So you have here the rate of uptake by diffusion and the rate of uptake by endocytosis. And as you can see, it is slower. And finally, very importantly, the antibiotic, which is taken up by endocytosis, will remain confined in the acidic compartment. It will only leave this vesicle very slowly by a process of exocytosis. So as I said, the good example for that is aminoglycosides. And finally, the last mechanism is what we call diffusion segregation. This mechanism will be used by molecules that share a very specific chemical property, which is a weak basic character. So in other words, these are molecules which possess an aminated function, and this aminated function can be protonated in an acidic compartment. And the typical example for that is macrolide. So what will happen for such a compound? So again, they will enter the cells by simple diffusion, and they will also diffuse in the acidic compartment. But once they are inside this acidic compartment, because of the acidic pH, the aminated function will be protonated. And of course, a molecule which is positively charged is much less diffusible than a molecule which is globally neutral. And therefore, the molecule will have only a very low tendency to leave this acidic compartment. Therefore, we say that they are segregated in the acidic compartment. So what can we say about this process? So this process will allow for a rapid and high accumulation, as illustrated here. The preferential accumulation will take place in the acidic compartment, and the release will be globally very slow. Okay, so this was a summary of the main mechanism. So based on that, what do we know about the antibiotics that we have in our arsenal today? So first of all, regarding diffusion, we know that all beta-lactams will use this process to enter inside the cell. So the accumulation is fast, but as I said, they do not accumulate. They will reach inside the cells an accumulation factor close to 1, so meaning a cellular concentration equivalent to the extracellular concentration. Fluoroquinolone also enters cells by diffusion, so this is also a fast process, but as you can see here, they will accumulate inside the cells. So how do we explain that? So at this stage, it is still unclear, but our hypothesis is that fluoroquinolone probably do bind to some cellular constituent, like some proteins, for example, that will retain them inside the cells and therefore allow for their accumulation. Regarding the drugs entering cell by endocytosis, so as I said, they will be confined in vacuole, and they are mainly represented by aminoglycosides, so for which the uptake is slow and the accumulation is rather low, and also by glycopeptides, for which the accumulation is also slow, but as you can see here, the accumulation factor can become very high, for example, extremely high for orithavancin, so the accumulation inside the cells can be as high as 100 to 300 times the extracellular concentration, and this is due to the fact that it will enter the cell by a adsorptive endocytosis, and therefore it will first stick at the cell membrane before being taken up by endocytosis. Third mechanism is diffusion segregation. As I said, this is best described for macrolide, and as you can see here, this allows them to accumulate to quite high levels, so between 4 times and more than 50 times. So the higher accumulation is observed for drugs which have usually more than one aminated function, because these drugs will become bicationic, so possess 2 positive charge, and therefore they are even more retained in acidic compartment. Another drug which will also follow this mechanism is radesolid, a new oxazolidinone in development. And finally, we have a series of other classes of drugs for which we know more or less the accumulation factor, but for which we have no clue so far regarding the mechanism by which they enter the eukaryotic cells. So let's now move to a few examples about the importance of these parameters for drug activity. So this first example will illustrate the importance of the subcellular distribution of the antibiotic. And for this example, I use 2 drugs, so the fluoroquinolone moxifloxacin and the glycopeptide orithavancin. So we looked at the activity against 2 bacteria, so Staphylococcus aureus located in acidic vacuoles and Listeria monocytogenes located in the cytosol. So you can see here the activity, so this is the growth of the bacterium in the absence of antibiotic. And if you look first to moxifloxacin, you can see that it is quite active against both bacteria. And this can be explained by its subcellular distribution because it is a fluoroquinolone. It enters the cell by diffusion, so it is mainly found in the cytosol, but it is also able to redistribute in the acidic vacuole, explaining why it is also active on Staph aureus. On the other hand, if you look to oritavancin, you can see that it is also highly active on Staph aureus, but not at all on Listeria. And this is easy to understand. Oritavancin enters the cells by endocytosis, so it is confined in the acidic vacuole and has no access at all to the Listeria, which is located in the cytosol. So this shows you that the antibiotics really needs to have access to the infected compartment to exert its activity. A second example is the role of efflux pumps. And I will show you here that we can increase the accumulation of antibiotic by inhibiting efflux, and therefore we will also increase its activity. So I take here the example of macrolide, azithromycin, which is a substrate for P-glycoprotein. And so if you inhibit P-glycoprotein using verapamil, for example, you will expect to increase the accumulation of the drug and therefore also possibly its activity. And this is illustrated here. So this is the accumulation of azithromycin in control condition, so about 25 times in these cells. So what we did in this experiment is that we also separated the different fraction of the cells in a soluble fraction corresponding to the cytosol, a granular fraction corresponding to the different organelles, and a nuclear fraction which contains the nuclei but also the unbroken cells. And so, as expected, you can see that azithromycin is mainly localized in the vacuoles, but you have also a small proportion in the cytosol. If you inhibit the pump using verapamil, you can see that you drastically increase the accumulation to 70 times. And interestingly enough, you can see that you increase the accumulation in a similar proportion in the vacuole fraction and in the cytosol. And therefore, you can expect an increase of activity in both compartments. I just show you here the effect on Staph aureus, so the bacterium located in the vacuole. And indeed, you can see here the activity for azithromycin alone, and here for azithromycin plus verapamil. And indeed, you can see that you make verapamil active at lower concentration, which corresponds to an increase in its intracellular potency. A third example is that you may try also to modify the subcellular distribution of a drug in order to make it active on a bacterium to which it is not supposed to have access. And this is an example with aminoglycosides. So I told you before that they are confined in the lysosome because they enter cells by endocytosis, and therefore they are not active on a bacterium like Listeria. So this is illustrated here. So you can see here the intracellular growth of Listeria in the absence of antibiotic, and you can see here that gentamicin alone is not active at all. But it is possible to do some specific formulation. For example, in this work, we did a formulation as nanoparticle for gentamicin that we combine with a surfactant and a specific compound. And so by doing this, we were able to modify the distribution of gentamicin. So you can see here the distribution of gentamicin alone. So the accumulation is slow, and it is mainly found in the granular fraction, so in the organelles of the cells. With this specific formulation, you can see that you markedly increase the accumulation of gentamicin, but you can see also that you get a high proportion of the drug in the cytosol, here the white part of the graph, while it was completely absent of this fraction for gentamicin alone. And as a consequence, you can see that for this formulation, you get also some activity against intracellular Listeria. So this means that it is possible to modify the subcellular distribution of a drug in order to get activity against specific bacterium. And finally, I will illustrate the importance of the intracellular pH for the intracellular potency or the intracellular MIC of an antibiotic. So a few years ago, we made a very surprising observation, which is that MRSA, so methicillin-resistant Staph aureus, were as susceptible as methicillin-susceptible Staph aureus to beta-lactams when they are intracellular. So this is illustrated here. So you can see the activity of meropenem and cloxacillin against an MSSA in white and an MRSA in black. And you can see that you have the nice dose response curve as we see before, which is exactly the same for both bacteria, even if, of course, the MIC shown here for MSSA is much lower than the MIC for the MRSA. So how can we explain that? In fact, we did an experiment in broth, and we compared the activity of this drug in broth at neutral pH and in broth at acidic pH. For the MSSA, as you can see here, there is no difference. The activity is globally the same. But if we move to the MRSA, you can see that you have no activity at all, or at least activity at very, very high concentration when your broth is at neutral pH, but that your curve is shifted, markedly shifted to the left, so meaning much more active at acidic pH. And this may explain, therefore, why we get activity intracellularly, because Staph aureus is in an acidic environment. So how can we explain that? So we did some circular dichroism experiment. I will not enter in the detail, but the idea is that at neutral pH, the conformation of PBP2A, which is the protein responsible for the MRSA character, is closed, and therefore the beta-lactam cannot interact with the protein, while at acidic pH, the conformation of the PBP2A will change, it will open and allow for an interaction for the antibiotic, which will recover its activity. Okay, so these were a few examples showing you how we can play a little bit with pharmacokinetics to try to improve activity against intracellular bacteria. And we will now move to the last part, which will consider cellular pharmacodynamic parameters that are predictive of efficacy for antibiotics. So first of all, let's look at the importance of the bacterial responsiveness. So of course, we can have some difference depending on the bacterial species we are looking at. So I show here again the comparison between Listeria in the cytosol and Staph aureus in the phagolysosome. And here we look to the activity of three different beta-lactam. But let's just focus maybe on ampicillin. And you can see here that ampicillin is in fact highly active on intracellular Listeria. And this is quite surprising because if you remember, at the beginning of the presentation, I told you that usually antibiotics are less active against intracellular bacteria than against extracellular bacteria. But here we have a clear exception. So Listeria is much more sensitive to beta-lactams when it is intracellular versus extracellular. To the opposite, if we look to the activity against Staph aureus, you can see the classical picture where the efficacy is much lower against intracellular Staph aureus as compared to extracellular Staph aureus. So the question here is, why are beta-lactams much more active against intracellular Listeria? So to try to explore that, we did some proteomic analysis in which we compare the profile of expression of protein of Listeria when intracellular as compared to Listeria in growth. And we observed a lot of change, of course. I will not go into all the details, but what is very important here is that we observed a decrease in the presence of protein involved in the synthesis of peptidoglycan. And therefore, of course, it makes sense. So we can imagine that the bacteria, so Listeria, when intracellular, considers that having a very thick cell wall is not useful to be protected because it is already protected by the cells, and therefore it decreases the synthesis of its cell wall. And of course, you know that beta-lactam acts upon the cell wall, and therefore, if you decrease the presence of the cell wall, you will favor the activity of the antibiotic because the cell wall is much more easy to destroy. A second type of consideration in this field is also that for a single bacteria, we can have different type of phenotype. And this is illustrated here for Staph aureus. You can have a normal phenotype, so with typical colonies, but you can have also what we call a small colony variants, which are, as you can see here, very, very tiny colonies, characterized, of course, by a reduced growth rate. And this is due to specific defects in the metabolism of the staphylococci, which become dependent for their growth of the presence of some specific nutrients like menadion, amine, or thymidine. So in this experiment, we look to the activity of a large array of antibiotics from different classes on a normal phenotype and a stable small colony variant of the same strain. So this is the same strain, but we have the normal phenotype and the stable small colony variant. And you can see that for most of the antibiotic, the activity is drastically reduced against the SCV as compared to the normal phenotype, with maybe the exception of oritavancin, which is as active against both bacteria. And this is quite understandable because we know that most of the antibiotic need to act upon bacteria that are dividing. And so if they divide more slowly, of course, they will be less susceptible to the antibiotic. So now the question is, what can we do to try to improve the activity of antibiotic against these intracellular bacteria? Because as you have seen, in most of the cases, the efficacy is really markedly reduced. So one idea could be to try drug combination. So there are a series of ways to study drug combination, and so I will just show you one here, but there are many other ones that you can use. And this one is called the FME approach. So FME is for fractional maximal efficacy. So what do? So you do a dose response as you have seen before for the two antibiotics you are interested in. So I show you here the curve for one of them. And then you can calculate specific concentration that will generate a specific fraction of your maximal effect. So your maximal effect is here. You can calculate it from your dose response curve. And then you can calculate, for example, the concentration here, giving 90% of the maximal effect, 70%, 50%, 30%, or 10% of the maximal effect. And you do that for drug A, and then you do that for drug B. And then you will combine the drug in order to obtain always the maximal effect. So this means that you will, for example, combine the concentration of drug B giving 90% of the maximal effect with the concentration of drug A giving 10% of the maximal effect. And you do like that all over the range of concentration. And then, of course, you do the experiment and you expose your intracellular bacteria to these different combination of drug. And then you can draw this type of graph where you have the concentration of drug A and the concentration of drug B. This is the fractional effect of one drug alone and the fractional effect of the other drug alone. And of course, if you have a simple additivity of the effect based on the calculation you did here, you should have a fractional maximal efficacy of 1 in all cases. Then, if you plot your experimental data, if your value is higher than 1, it means that you have a synergy because the experimental effect is higher than the simple additivity. If your experimental point is below this line, it means that you have either indifference or even antagonism. So this is a little bit complex, but here is an example, an experimental example for combination against small colony variant of Staph aureus. On the left, you have the combination of rifampicin and moxifloxacin. Here you have rifampicin alone, you have moxifloxacin alone, and here you have the combination. And you have understood now that this combination is only additive. But if you look to the combination of rifampin and oritavancin, you can see here that you have a huge synergistic effect. And therefore, we may suggest that using a combination of these two drugs may help getting a better efficacy against intracellular Staph aureus. And finally, the last example I will take is that of the cooperation with host defenses. And for that, let's go back to Listeria monocytogenes, and we will look to the influence of interferon gamma, which is one of the cytokines that play a role in defense against the infection. So first of all, let's look to the activity of antibiotic alone. So this is the control, so the intracellular growth of Listeria. This is ampicillin alone, azithromycin, a macrolide, and sparfloxacin, a fluoroquinolone. So all of them show some sort of activity against intracellular Listeria. So remember that Listeria is found in the cytosol. Then, if we add interferon gamma, what do we observe? So first of all, we observe that the localization of the bacteria has changed. So now you can clearly see here that the bacterium is located in a vacuole. So what are the consequences of this addition of interferon gamma for activity? Well, for ampicillin, you have almost no change. But for azithromycin, you have a slight increase, and for sparfloxacin, you can see that you have a huge increase in its activity. So the question now is, why do we have this increase of activity? Is it related to this change of localization? So to answer this question, we can use a control consisting in incubating the cells with catalase and N-name. And so these two molecules will in fact destroy the oxidant species that are produced in the macrophage under the influence of interferon gamma. So if we do that, you can see that the bacteria are still in the vacuole, but if you look to the activity of the antibiotic, you can see that it comes back to the control level. So this experiment shows that this is not the localization of the bacteria which explains the increase of activity, but rather the cooperation between the antibiotic and the oxidant species that are produced by the eukaryotic cells when it is exposed to interferon gamma. Therefore, these experiments show that intracellular antibiotic can clearly cooperate with host defenses to eradicate bacteria. So we are now coming at the end of this presentation, and to conclude, I try to establish a sort of wish list of the characteristics we may try to have if we wish to develop a new antibiotic active against intracellular bacteria. So if we look to pharmacokinetic properties, I think that we need to develop antibiotic which can have access and possibly also to accumulate in all subcellular compartments in order to be able to act upon any type of intracellular organism. Preferably also, we need to develop antibiotics which are not substrate for efflux by eukaryotic pumps in order to remain inside the cells for a sufficient period of time. Then we need also to develop antibiotic showing activity at both acidic and neutral pH, because we have seen that the acidic pH in the phagolysosome can markedly modify the activities of some antibiotics. However, so all of these pleads for an accumulation inside the cells, but we have also to consider that if we have an antibiotic accumulating to high level and also remaining for prolonged period of time inside the cells, we may have some risk of toxicity. So I didn't have the time to speak about that today, but it is certainly something we have to take into consideration. Regarding now pharmacodynamics, we need to try to have antibiotics that can express their activity intracellularly, including against slow-growing phenotype, because these are probably a main reason for persistent infections. Possibly also, we need to have antibiotics that cooperate with host defenses. And finally, we may like to consider combination, including with agents that can increase bacterial responsiveness. So I didn't speak about that today. I limit myself to antibiotic, but there are also some agents that can increase the bacterial responsiveness, for example, by favoring the growth of slow-growing phenotype. And so with this, I think I'm at the end of my presentation. I would just like to finish by acknowledging my colleagues over the years in the lab who did the experiment that I presented today. I wish also to thank Professor Paul Tulkens, who was my previous supervisor. And I wish also to thank my financial support in Belgium, so the Belgian Science Policy and the Fonds National de la Recherche Scientifique, which supported my research over the years. So I thank you very much for your attention, and I will be happy to take any question.
Thank you, Françoise, for your excellent presentation. We will now start the question and answer session. As a quick reminder to the audience, please would you send your questions via the window on your webinar control panel, and we will do our best to respond to as many questions as possible in the time remaining in this webinar session. So our first question is, are some antibiotics activated intracellularly?
Okay, I'm not sure what the person means exactly by activated. We may think, for example, and we did some work a long time ago with that, of using some prodrug. Maybe this is the question. So you can indeed use sometimes some prodrugs that may possibly diffuse more easily through the cell membrane, and this can be the case, for example, for some products of beta-lactams. So this will increase their capacity to enter inside the cells, and then of course this prodrug can be destroyed and regenerate the active antibiotic inside the cells, and therefore the antibiotic will be able to exert its activity. So this is the main point I'm seeing about activation. I think that some people also suggested that antibiotic like fluoroquinolone, for example, could possibly form radical species, and so possibly this could be the case if you have a very highly activated macrophage producing a lot of radicals, for example. So for me, I think these are the main example I can give for this type of activation.
Thank you. The next question is, you've clearly given a lot of data showing the number of bacterial cells that are either intracellular or extracellular. Do you also measure the amount of drug that's in the cells as well?
Yes, of course. This is something which is very important to do. So I didn't show experimental data about accumulation. So I just give you a final overview of the accumulation factors for these different drugs. But of course, this is something which is very important, which is to measure so not only the accumulation, but also the subcellular distribution. So to know where your antibiotic is. So is it in the cytosol? Is it in the phagolysosome? So this may be very important in terms of activity against bacteria located in one or the other compartment. So to measure the accumulation, so what you need to do is to incubate your cells with your antibiotic in the condition that you are interested in, so in terms of concentration or of time of incubation. Then you have to wash your cells to eliminate the extracellular antibiotic. So it's very important to do this step at 4 degrees C to avoid, of course, that the antibiotic could leave the cells during the process. And then you can lyse the cells and measure the antibiotic by an appropriate technique. And so this technique, of course, can depend on your antibiotic. If you have a radiolabeled drug, it's of course very easy, but at the present time, it's quite also easy to develop mass spectrometry method coupled to HPLC in order to assay the drug concentration. And then, as I said, the other important point is to also evaluate the subcellular distribution inside the cells. And so for that, usually what is done is to fractionate the cells using centrifugation. So for that, you will in fact centrifuge your cell extract at a different speed. And so at low speed, you will pellet mainly the nuclei, and then if you increase the speed, you will pellet the organelles, and of course the remaining part is the cytosol. And then you may have more sophisticated technique, allowing also to separate one organelle from the other one if you are interested in determining in which organelle is located the drug more precisely. So this is a classical biochemical techniques. Now, of course, there are also other techniques. So, for example, if your drug is fluorescent, you can also have a look in the confocal microscope, for example, to see where is the fluorescent signal. So, of course, this is another possibility. But indeed, I think it's very important to look in parallel to pharmacokinetics of the drug and activity in order to correlate both.
Thank you. Speaking about the cooperation with the host, did you also study antibiotics which reduce the host defense?
No, this is something we have not done, but of course this is also something which could be very important because the interaction can go both ways, and so you can have positive interaction and also negative interaction, and there need also, of course, to be taken into account in your drug development process because this may be deleterious for the patient, of course. If you reduce the host defense, you can also make the patient more sensitive, possibly to other infection, and possibly also if you can have some resistance, you will have some difficulties to eradicate the bacterium.
Thank you. Are there any intracellular models for the treatment of gram-negative bacteria?
Yes, of course. So here I just took two examples to avoid going in all directions, but there are models for gram-negative intracellular bacteria. Salmonella, for example, Shigella, Escherichia coli, and other bacteria, even Pseudomonas. In some condition, you can keep it intracellularly. So it's always a matter of equilibrium between the amount of bacteria and the amount of cells, as I said. So if you have bacteria producing some toxins, of course, if you have a too high inoculum, you will kill your cells. And of course, in this case, you will not have any more intracellular infection. But if you use an adapted inoculum, you can maintain them inside the cells.
Thank you. When investigating combinations of two drugs, how do you manage when you have drugs with very different pharmacokinetic profiles?
Yes, that's a very good question, because the experiment, as I show you, is an experiment in which we expose, in fact, the infected cell to, let's say, a continuous concentration of the drug, so that we did not mimic pharmacokinetics in this type of experiment. If you have two drugs with different concentration, with different pharmacokinetics, and if you wish to take that into account when evaluating the effect of the combination, I think you would need to develop much more sophisticated models in which you use a system with pumping, which allows you to readapt the concentration of the drug over time. So this is certainly the best solution, but it is probably very complex. Now, another possibility I'm thinking about is also that we have now more and more powerful population pharmacokinetic models, and so you can probably do experiment in static model and then combine them with population pharmacokinetic data in order to use your in vitro data and incorporate them in the pharmacokinetic model to try to calculate what would be your effect when your intracellular bacteria are exposed to varying concentration. But again, this would require an expertise in population pharmacokinetics.
Thank you. Are there in vitro models that use fluctuating concentrations versus static concentrations?
Okay, so this is part of what I said before. So they are not very common in the literature, but I think that some people start to develop this type of thing. So it's like a hollow fiber model that is classically used for extracellular, for evaluating extracellular activity of antibiotics. So you have to adapt this type of hollow fiber model in order to incorporate infected cell in the system rather than simply bacteria.
Thank you. If rifampin was to be delivered into lysosomes, would it be stuck there or will it be released to kill bacteria in the phagolysosome?
Could you repeat, please?
If rifampicin was to be delivered into lysosomes, would it be stuck within the lysosome or will it be released to kill bacteria in the phagolysosome?
Yes. So, as I said, at the present time, we do not know exactly how rifampicin is taken up by the cells, neither in which compartment it is going by itself. But I guess that the question here is that what would happen if we prepare a sort of vector or formulation allowing the rifampicin to reach a specific compartment. So in this case, it will depend, of course, of the diffusibility of rifampicin from one compartment to another one. So if you develop formulation, for example, driving it to a specific compartment, you may have some drug in other compartments if it is able to redistribute, meaning if it is able to cross the membrane inside the cells. And so I suppose it should be the case because, as I said, rifampicin accumulates to quite high level by itself. And so I think that it should show some diffusibility and therefore probably some redistribution inside the cell. But this is something which need to be tested. So we also know that it is quite active intracellularly against bacteria like Mycobacteria. But but also against Staph aureus, and these two bacteria are already in different compartments. So it means that it has at least access to these two compartments. So I think it's all what I can say on that for the moment.
Thank you. Are there models for anaerobic intracellular bacteria?
Again, that's a good question. I think there are. I think there are, but of course it makes again things much more complicated, because you have to cultivate them in an anaerobic atmosphere when they are extracellular, but eukaryotic cells need to be, of course, in an aerobic atmosphere. But I think there are some models with bacteria like Coxiella, for example.
Thank you. Turning to inhibitors of efflux in bacteria, do these have any role in altering the intracellular infection? So inhibitors such as those of the ACR-AB and TOL-C proteins in bacteria?
Yes, that's a very good question. Yes, indeed, they have a role. So we did some experiment, not with gram-negative, but we did that with Listeria, and we compared the activity of fluoroquinolone against Listeria that express or do not express efflux pump to fluoroquinolone. And indeed, we observed a marked difference in activity. So meaning that the efflux pumps was clearly expressed also intracellularly. So as I said, this was for Listeria. So if the same takes place for gram-negative, and if these efflux pump like Acrabate also are also expressed intracellularly, it should play a role. We did also some experiment with Pseudomonas expressing or not MexAB OPRM, for example, which is homologous to Ecrebi TOL-C. And indeed, we saw a difference in the activity of antibiotic. So usually what you observe in this case is rather a change in potency, so meaning that if the bacterium is expressing pumps, you need a higher concentration of antibiotic to get the same effect, but you will not observe a change in the efficacy, meaning that you will always get the same maximal effect. So this is clearly a pharmacokinetic related parameter, which is logical for an efflux even in the bacteria.
Thank you. During drug discovery and exploratory research to help choose candidates to go through into clinical studies, where would the experiments that you've described today fit into that development program?
Okay. I think it could come quite early, in fact. So of course it depends on the bacteria you are targeting. So if the bacteria you are targeting is clearly intracellular, I think it should come very early on in your preclinical phase of development, because it makes no sense to do a lot of experiment against extracellular bacteria if the life cycle of the bacteria is mainly intracellular. Now, we have also a lot of bacteria which are considered as facultative intracellular, like Staph aureus. So for this one, let's say that the intracellular life is one possibility, but of course you have also a large part of the pathogenicity which is related to the extracellular life, and so for this maybe it's a little bit less urgent. But anyway, I think I would include it in the early preclinical phase, because it's also quite important in terms of looking how your antibiotic will interact with eukaryotic cells. Will it be able to enter inside the cells? Will it exert some toxicity against these cells? And these are types of information I think that you may need quite early, because if you have, of course, a major toxicity, it makes no point to go further with animal studies, for example. So I remember one drug that we tested in the lab a long time ago, and so in fact we observed a gigantic activity against intracellular bacteria that we never saw before. We say, oh God, this drug is really very active. But in fact, we looked to the cells in the microscope and there were no cells anymore. In fact, this drug was highly toxic. And so what we were measuring, in fact, was extracellular activity. And so I think that this is a useful piece of information you can include early on in your development, because anyway, even if this is a bacterium which is partly intra and extracellular, it is very useful to know what would be the consequence of the interaction of your drug with the eukaryotic cells.
Thank you. If you had a drug that had only a slight negative charge, where would it be most active? In other words, where would it have its greatest antibacterial activity?
So a drug which is negatively charged,
But only slightly.
Yes, because I'm thinking about a drug which is negatively charged. So in fact, most of the antibiotics are either zwitterionic, so meaning they have both a negative and a positive charge, or they are cationic, like macrolide, and so we have a few exceptions. So beta-lactam have a carboxylate function, but at physiological pH, this function will be protonated. So there will not be an anionic, and so maybe there are some fluoroquinolone, some new fluoroquinolone like delafloxacin, which is anionic. And so, in fact, what will happen with this type of molecule is that because of this negative character, the diffusibility inside the cells will be a little bit reduced. And but if you put the cells in an acidic environment, which is the case in many infected territories, in fact, this anionic drug will also become protonated and will become neutral. And so in this case, its diffusion inside the cells will become even more important than for a zwitterionic one.
Thank you. If you had a drug that was within a membrane vesicle, would this be delivered into this inside of a cell? And do you have any experience of this?
So I suppose that the person is referring to drug entrapped in liposomes or any type of vector like that. So yes, of course, but you have to develop your vector so as to allow its entry inside the cell. So you may have different type of these vector systems. So you have some of them that will be taken up by phagocytosis. And so in this case, you are expecting that the drug will be delivered in the phagolysosomes, but you have other ones which can also fuse with the pericellular membrane, and so in this case you will deliver the drug in the cytosol. You have other ones that can be activated at acidic pH, for example, and so once they are in the lysosome, they will again deliver specifically the drug in this acidic vacuole. So I think there is no one response. It will really depend on the type of formulation you will use and of its properties.
Thank you. And this is your last question, Françoise.
Okay.
Verapamil will have side effects since it affects calcium channels. Are there better drugs that work in a similar way to verapamil, but without the side effects that you've tested?
That's a good question. So indeed, verapamil is classically used as an inhibitor of PGP in the lab, and I would say this is the same for other pumps. So of course, you cannot use it in a human for inhibiting PGP because the concentration needed to inhibit the pump are much higher than the one that have cardiovascular effects. So you will certainly intoxicate your patient. So that's not a good solution. The problem now that we have is that several companies try to develop inhibitor of efflux, and this is true for eukaryotic pump, but this is also the same for bacteria pump. So different companies try to develop inhibitors of efflux, and for the moment, as far as I know, none of them came to the last stage of clinical development because of toxicity. So there was, for example, elacridar, if I'm correct, which was an inhibitor of P-glycoprotein. So this one went to clinical trial, but the clinical trial were stopped because of toxicity. So it was evaluated in combination with anti-cancer chemotherapy, but in fact it was toxicity. It was toxic, and I think it's quite easy to understand. In fact, you have to think about the fact that these efflux pumps are located almost everywhere in the body, including to protect the brain, so at the blood-brain barrier and so on. And so if you have an inhibitor inhibiting transporting at the level of all the barriers of the body, it would be really difficult to avoid having toxicity. So it's why on my last slide, I suggested that probably a better solution would be to screen again quite early in the development process whether the drug itself is substrate or not for efflux. And if you have two drugs, let's say, which are equivalent in terms of activity, but not one is substrate for efflux and the other one is not, probably it's a better idea to develop the one which is not substrate for efflux, because you will simplify the pharmacokinetic profile.
Thank you. So we've now come to the end of our question and answer session. I want to thank everyone in the audience for contributing to this discussion with their questions, and I apologise to those for whom we've run out of time and so your questions could not be answered. And of course, I want to thank Françoise for her time and for sharing her expertise with us. Goodbye, everyone, and I'm handing over to you, Astrid.
Thanks, Laura. I would also like to thank Françoise for her presentation and for her very insightful comments during the Q&A. I would now still like to take the opportunity to announce another webinar in July, as well as our August webinar. On the 18th of July, Ursula Teuratsbacher will give again her presentation, Enabling Academia to Fill the Discovery Gap, Learnings from Industry and Funders. This presentation was part of a Meet the Expert session which GARDP co-organized together with Carbex and the Repair Impact Fund at the last ASM Microbe Conference. On 20th of August, Lena Friberg and Elisabeth Nielsen from the Uppsala University in Sweden will present in a webinar on computational modeling for population PK and PK/PD. You can already register for these webinars on Revive under revive.gardp.org/webinars. And with this, I would like to thank you all for joining today and for contributing to the discussion. I really hope this webinar was interesting and useful for you, and that you will join us again for our future webinars. Please make sure to spread the word in your networks and to encourage your colleagues to join as well. Thank you and goodbye.